Hi, this is Jiyue from Washington State University. Today, I would like to talk about our effort to generate a mouse model with humanized telomere. We know that human cells have a limited lifespan. If you culture normal human cells, they will proliferate and eventually reach a state of the plateau. And this is called the replicative senescence. This was initially reported by Dr. Hayflick. It's 60 years ago. And so it's also called the Hayflick limit. So over the last 6 decades, there has been extensive research on understanding of this replicative senescence or replicative aging. So now we know that telomere shortening is a fundamental mechanism which induces this senescence. As this telomere shortens, they lose chromosome protecting function, leads to chromosome DNA damage and apoptosis and senescence. So replicative senescence is not conserved among mammals. And we humans have relatively short telomere, between 10 to 15 kb of telomere. And so we have telomere-dependent senescence. On the other hand, many mammals, including mice, have very long telomere, and they do not have telomere-dependent senescence. So this raises a lot of questions. So while mice have long telomere, are long telomeres artifact of laboratory breeding? So we know in addition to long telomere, mice also express telomerase in adult tissues, and whereas in human, telomerase expression in tissue is very low. So our hypothesis is that repression of the H2 gene during development leads to short telomeres in human. So in order to test this hypothesis, we spent many years trying to understand how the human TER gene is regulated. So now we know there are several elements are required for TER regulation. The promoter is important. The promoter contains the binding site for many transcription factors. And we know that the promoter is important for both activation and the repression of the H2 gene. And we also know that the intron 2 has a critical for TER regulation, specifically the VNTR2-1 we identified a couple of years ago is a enhancer of the TER gene that's required for TER expression in cancer cells. And intron 6 is also important for both transcription and splicing of the TER gene. So in addition to these elements, there are abundant repetitive elements within the H2 locus. Compared to the mice, there's much more of these sequences. And these sequences, mainly transposable elements, are important for establishing a repressive chromatin environment that's responsible for silencing of the H2 gene in somatic cells. So based on this knowledge, we designed a strategy to construct a humanized mouse telomerase gene. And we did so by replacing the 5' intergenic region intron 2 and intron 6 with the human counterparts. And this strategy does not affect amino acid sequence of mTOR protein, because we know that H2 protein does not function in mice. So we made a construct, targeting construct, because the region for modification is very long, and it's about 50 kb. So we had to use CRISPR cleavage to help the homologous recombination. And we successfully achieved homologous recombination in mouse embryonic stem cells. And the right side slide shows you how we got this correct clones with modified HM3 gene. So the ES cells were injected into the albino host of C57 black mice, and the chimera mice was obtained as shown on the right. So with these mice, we crossed these mice to the homozygous TER knockout mice to generate the mice with HM3 allele and M3 allele. So if you can see here that So we look at the telomerase expression in adult tissue. Showing red are the mouse with the mTOR gene, and as you can see, this mouse express telomerase in most of its adult tissues. On the other hand, its sibling with the HM third allele express the highest level of telomerase in thymus and low level in testis. So we further characterize its expression by using QRT-PCR, as shown on the right. That this is done using the heterozygous mouse with one HM third allele and one third mTOR allele. The shown in red are the wild type mTOR gene, and as you can see, it's expressed in most of the adult tissues. On the other hand, the HM allele was expressed mainly in thymus and also low level in testis and ovary shown here. And the lower side slide shows you the H3 expression in human tissue. As you can see, the HM3 gene recapitulates the regulation of the H3 gene in human. So these mice still have very long telomere. So in order to shorten the telomere, we utilize the turtle knockout mice. And it's reported that the turtle knockout mice can survive for up to 6 generations. And with each generation, the telomere length shortens. So we cross the heterozygous H minus and the plus minus to the successive generation of turtle knockout mice. As you can see here in the middle, showing you the flow fish measuring telomere length. The flow fish is method for using the in situ hybridization using fluorescent labeled telomere probe. The data is normalized to wild type C57 black mice, which has about 50 kb of telomere. As you can see, with each crossing, each generation, telomere shortens progressively. And at the generation 6, and we reached about 50% of wild type telomere length. The right side slide shows you the telomere restriction fragment analysis, basically confirming the data we obtained using flowfish. So these mice also have maintain their body weight. In fact, the turtle knockout mice have significantly reduced the smaller body weights compared to these mice, and the heterozygous mice maintain their body weight in both male and female mice. So testis is an organ that's is very sensitive to telomere dysfunction, because the cell proliferation occurs throughout the life in this organ. So as you can see, in the late generation mice, G4 and G5 mice, there is significantly reduced the testis weight. However, in both H minus and plus minus, the testis weight maintained at the normal level. And on the right side shows you the HNE staining of testis. These structures are seminiferous tubules. As you can see in the knockout mice, late generation knockout mice, there are extensive loss of germ cells in these tubules, whereas you don't see this happening in the H minus mice with HM allele. So we further look at the lifespan of these mice, and we show here that the G4 and the G5 mice have shortened, reduced the lifespan. But the mice with one allele of either H3 allele or HM3 allele have lived well beyond 500 days of this experiment time. So this data indicated that H2M3 allele is a functional gene that maintains tissue renewal in mice. So as I said earlier, that the turtle knockout mice can survive up to 6 generations. And indeed, we only found one G6 mouse which died prematurely. This effectively terminated this breeding strategy. So we start to intercross the heterozygous H minus here, starting from G4 mice for 16 generations. And next slide shows you the telomere length using the flowfish data. And you can see the telomere length decrease each generation, and eventually stabilize in the last 3 generations mice. And as you can see, these mice maintain the body weight and the testis weight. So here we show the telomere length of these three genotypes. As you can see, the HH homozygous mice tend to have longer telomere than both the heterozygous mice and the knockout mice, and indicating that the HM13 is haploid insufficient. And throughout most of the breeding process, so we found that 50% of mice born are heterozygous H- mice, and the homozygous HH and the minus made up made about 25% of total progeny. This follows the Mendelian genetics. However, in the last three generations, we found that there are precipitous decline of number of homozygous knockout mice, and those mice born are actually die prematurely. So this indicating that the short telomere in these late generation mice cannot sustain mouse development without the functional gene. And finally, we also want to visualize the telomere in these mice. As you can see here, that this is the wild type C57 black 6 mice. These mice have about 50 kb of telomere. And these long telomere cannot be resolved in this 0.6% agarose gel. We also shown here the normal human foreskin fibroblast cell at the passage 11, and these cells have a telomere smear, as you see in many other human cells. And for the mice with the telomerase HM13 gene, we can see there are two types of telomeres. One is the distributed band between 15 and 20 kb. We believe that these are the ultra long telomere that has yet to be decreased to the human length. And we also see the telomere, human-like telomere smear. For the HH homozygous mice, the smear is between 8 to 10 kb, and for the heterozygous mice, it is between 6 and 8 kb. In total homozygous knockout mice, the telomere smear signal is dramatically reduced. So we also want to know if these telomeres are stable in homozygous mice. So we started the in-cross of HH mice. We established 3 lines, G4 line, G4.8 line, and the G4.14 lines. And their telomere signals or lengths are shown here. As you can see that with each breeding generation, and telomere length declines. And for the G4.8 and the G4.14 lines, telomere appears to stabilize around 20% of wild-type mice. And here is showing you the southern blot of telomere. And you can see this is the last 3 generations of G4 mice, and this is the last 3 generations of G4 and G4.14. And this, especially for G4.8 and G4.14 lines, the telomere smears average about 10 kb. This is very similar to the human telomeres. So these actually mice also maintain their body weight, and as body weight. So because the telomere deficiency, one of the major problems of mice and as well as human with telomere deficiency is the bone marrow failure. So we look at the peripheral blood of these mice. Here we show this is the wild type C57 black 6 mice, and this as negative control, which also show the G5 to the knockout mice, and the normal physiological data of C57 black 6 mice published by Jackson Lab are shown here on the right. So as you can see, the HH mice have normal red blood cell counts, normal hematocrit, and although there are some variation in white blood cell counts, but most all these data are within the normal range of C57 black 6 mice. And within white blood cell, we can see there have normal number of lymphocytes, normal number of monocytes, and the neutrophil. as well as in the among lymphocytes, we saw normal number of CD4 T cell counts, CD8 T cell count, as well as the ratio of CD4 to CD8 cells. Finally, we also see that the testes are normal, both weight and structure. And so we also want to know in the pathological condition whether the cell renewal capabilities affected in HH mice. And so we use the dextran sodium sulfate treatment to induce the colitis-like condition. And the DSS treatment of 6 days will lead to severe intestinal damage. And upon withdrawal of this compound, and there will be a compensatory cell proliferation, and that can be captured by EDU labeling. As you can see here is the EDU labeling experiment. This is the wild-type mouse, and this is the HH mice. And there's the significantly reduced the number of EDU positive cell in these HH mice compared to wild-type mice. And on the right side shows you the summary of the data. And as you can see that there are average 7 EDU positive cell in the wild-type mice compared to the only 2 cells per crypt in the HH minus. So these data indicating although the HH minus were normal under the normal physiological condition, but the tissue renewal was more limited under the pathological condition. Finally, we also look at the proliferation of the mouse embryo fibroblast cells. These cells are isolated from individual embryo and cultured at the 3% oxygen. And the wild-type MAF continually proliferate. The HH MAFs actually start to show the plateaus after 40 population doubling, and the H- mine heterozygous MAFs actually have reduced the number of population doubling, and there is a significant decrease of the cell proliferation capability in total knockout mice. And these data indicating that the MAF from HM, those mice actually acquired the telomere-dependent replicative senescence. So in summary, we have engineered the HM3 gene, and this gene recapitulates the regulation of human TET gene, and the short telomere result from these heterozygous mouse intercrossing becomes stabilized in the HH mice, and HH mice and maintained the human telomere, the human length, that's creating a strain of C57 black 6 mice with humanized telomere, and we called it HUT mice. Somatically, of these cells from these mice display limited proliferation potential and also acquired replicative senescence. So the HM3 gene appears to be haploinsufficient, and just like H2 gene, these mice can be used as a preclinical model for dyskeratosis congenital and as a telomere biology disorders. And finally, so these are the people who did the work at WSU, and the total knockout mice were from Yale and Leo Harridan's lab, and below are our funding sources. Thank you.